Bien, écoutez, bonjour à toutes et à tous. C'est un immense et réel plaisir que de vous accueillir à cette 29e journée d'endocrinologie, métabolisme et nutrition de Sorbonne Université. Alors, vous le savez, autrefois, ces journées s'appelaient les journées du CHEVALIER. La configuration géographique des structures hospitalières font que nous sommes tous regroupés, hôpitaux de l'Est parisien, au sein de Sorbonne Université. Et 29e journée veut dire que cela fait près de 15 ans que nous animons 2 réunions annuelles, toujours le premier vendredi de juin et le premier vendredi de décembre. Et puis, comme vous l'avez bien compris, depuis 2 ans, nous ne nous sommes pas retrouvés pour la simple raison du Covid. Et vous voyez même qu'aujourd'hui, au dernier moment, j'ai dû faire annuler le traditionnel buffet pour des raisons d'interdiction de la direction de l'hôpital pour éviter tout risque de contamination. En tout cas, mille merci à la centaine de personnes qui se sont officiellement inscrites pour être présentes à nos côtés aujourd'hui. Mille merci aux plus de 440 personnes inscrites à distance. Et j'en profite pour leur dire que s'ils veulent nous envoyer un petit message pour nous dire d'où ils sont, d'où ils viennent, c'est avec un immense plaisir qu'on pourra dessiner une cartographie de cette journée d'endocrinologie. Et je voudrais remercier tout particulièrement le comité scientifique de ces journées qui est animé par l'ensemble des universitaires de notre discipline en endocrinologie, diabétologie, métabolisme et nutrition. Alors la particularité de cette journée, c'est qu'il va y avoir évidemment différents exposés. Il va y avoir toujours cette fameuse saga des cas cliniques qui attire toujours beaucoup puisque ce sont des expériences de la vie quotidienne auxquelles nous sommes confrontés. Mais nous allons tenter une expérience un peu nouvelle, c'est-à-dire que puisqu'on est aujourd'hui dans du digital d'une façon très importante, vous avez, je suppose, tous sur vous un téléphone portable. Vous avez dans votre tote badge, si je puis dire, le programme de la journée avec un QR code. Et en flashant ce QR code, vous allez être mis en relation directement avec votre intermédiaire pour poser des questions, pour répondre à des questions qui vous seront proposées par les différents orateurs. Donc prenez quelques secondes pour flasher ce code et voir ensuite comment vous allez pouvoir répondre à ces questions. Et je vais en réalité maintenant lancer les débats avec 2 sessions différentes. Il n'y aura pas de pause buffet tout à l'heure, mais il y a une pause d'une demi-heure. Et c'est important pour nous de vous dire que tout cet événement, en plus dans sa dynamique hybride, digitale et présentielle, a été possible grâce à 2 acteurs. Un des acteurs, c'est la société Terra Médicale, avec Aurélien Rivet et Maxime Latry, avec qui on a travaillé, je pense, tous les jours, depuis 4 mois. C'est-à-dire qu'on a été très intensément impliqués les uns par rapport aux autres, mais ça a été un vrai plaisir et une vraie relation amicale qui s'est créée. Et puis je voudrais remercier tous les partenaires de l'industrie qui ont joué totalement le jeu, un peu au dernier moment puisque ça s'est organisé en septembre, et des partenaires qui ont décidé d'être des partenaires or, d'autres argent, d'autres bronze pour des raisons plus sans doute réglementaires qu'autre chose. Et on verra, on espère qu'en juin 2022, pour la 30e édition, on vous concoctera un programme encore plus beau que celui d'aujourd'hui. Donc, profitez de cet après-midi. N'hésitez pas à interagir avec les différents orateurs. Et je vous souhaite une très, très belle journée. Merci à vous toutes, merci à vous tous. Alors, je passe la parole, parce que tout ne peut jamais être parfait. Il y avait 2 modératrices, mais la deuxième va arriver incessamment sous peu. Mais pour l'instant, Camille Vattier de Saint-Antoine va lancer les débats et ouvrir la première session. Merci Camille.
Bonjour à tous, merci beaucoup Philippe pour cette invitation. On est quand même ravis de se revoir, même si c'est un peu un format hybride, mais on est tous habitués à ça. Et donc je suis très heureuse d'accueillir pour commencer cet après-midi le docteur Hilke Plassmann, qui est professeur à l'INSEAD en neurosciences sur la prise de décision, je pense qu'on peut le dire comme ça, et qui est également professeur associé dans le domaine du marketing, donc vous voyez une activité très mixte, et qui va justement nous parler de quelque chose de très nouveau sur les connexions dans le cerveau avec la régulation de la prise de décision et des comportements. Et donc le sujet, c'est les différences individuelles dans le choix et le contrôle de nos aliments, quand l'estomac et le cerveau se connectent pour mieux comprendre notre comportement alimentaire. Et donc on va revoir ces travaux sur les différents modèles qui existent pour essayer de mieux comprendre la prise de décision et en quoi cela peut nous apporter quelque chose dans le domaine de la nutrition. Voilà, donc merci beaucoup de votre présentation.
Okay, so I'm going to continue now in English. So as already Camille was saying, infrared outside from the Sanofi stand was saying, it's funny that you're here because I know INSEAD is a business school in Fontainebleau. That is true, but in the field of marketing or consumer psychology, which is my field, there are a lot of researchers that are studying how can we help consumers to make better decisions. And one area is nutritional decisions, so dietary choices. And this is what a lot of my research agenda is actually dedicated to. And so specifically, I'm interested in individual differences, sourcing not only from behavior, but as Camille was saying, also from the neuroscientific level. So I have a training in cognitive neuroscience. I have actually a research lab right next door at the ICM, so it was a short way to come here. And also, so this I've been doing since my PhD, but since 6 years, I also am in the learning phase of trying to learn more about microbiology. Luckily, I have awesome collaborators that help me to better understand this. So what is it that I'm interested in? So here you see actually my 2 children, 2 boys, and this is Mats. And so this was during COVID when I had I couldn't run any experiments, so I was starting to run experiments with my poor children. And so I put in front of him a bowl of chocobons and asked him, so look, if you can wait to eat any of these chocobons, you can actually keep them. But if you can't wait, you can only eat one, and I'm going to take away the bowl. So this is Mats. At this time he was 5 years, and you see he's actually very good. You know, he can wait and play a bit with this. This is his younger brother, Yannis. Yannis sees this, immediately touching it. I couldn't even take a picture without him not touching it, and boom, in his mouth, right? So, and this means the two are very different with respect to their ability to delay gratification. In fact, all of you that have children are doing field experiments at the moment when you think about the advent calendar filled with chocolate, because this is also a delay of gratification task. And Yannis, my youngest, who's now 3, he ate all of the chocolates in his calendar the first day the calendar was out. Right? So whereas Mats is still very good at not doing this. So there's something, and then obviously here there's a difference of 3 years in age between my children, which could be one factor kicking in here, because we know actually that in adolescents and children, the delay of gratification is less pronounced, right? But the ability, so individual differences, and this has been shown to predict outcomes later in life, such what jobs they have, and so forth. But so these differences, I'm not interested in the developmental angle. But this is a paper that was one of the first papers that looked at this from a neuroscientific angle, who looked not at chocobons or marshmallows, but at monetary rewards. So the choice between a smaller sooner versus a larger later monetary payoff, right, which is important for retirement savings and so forth. From this paper, what you see here is that, so this is a way to look at how much people are discounting, right? So the steeper the curve, the more they are discounting. And you also see a lot of individual differences. However, this has not been studied yet in that extent, and this is what I'm interested in, is to understand why people are discounting at different rates, different types of rewards. And so I'm specifically interested in these now versus later conflicts. In the food domain, I also show you some things in the monetary domain, which is in some ways a bit easier to study because the food is a bit more difficult to study, because in these experiments you really have to give it to the participants. But then there's also something that the psychological literature thinks of as a similar trade-off between a short-term versus a long-term reward or goal, which is the idea of, I say, Fabrizio, do you want to pick the chocolates now versus the Brussels sprout later. Right? So the Brussels sprouts are the long-term reward because, as all of you know much better in this room, it's good for your long-term health. Whereas this one is good for his long-term reward because he likes eating them, but maybe less good if he does this repeatedly for his health, right? And so these are the types of decisions I'm going to talk about in this presentation. And I will first start with telling you a bit more about what we know on the neuroscientific level. And then I'll show you a bit about what's actually happening at the microbiological level in the gut, given that this is a good fit with the topic of this research day. Voilà. So I'll start with a couple of studies that I've done that are looking at the brain signatures of this behavior and the role of individual differences on the brain level. The first study was joint work with, so the first author, my former postdoc, Liane Schmidt, and then there's Sandri Hutchins, Anita Tush, and Todd Hare from various universities. And here we had a similar task to the task I just showcased with Fabrizio, where we asked participants, so this is what they saw in the scanner, I want you to make decisions on these different food items, and you can reply strong yes, no, yes, strong yes. And in some of the decisions, I asked them to really focus on the healthiness of the items. So I want them to make them healthy decisions. In others, I asked them to really focus on the tastiness of the items. And then some I asked them, just make decisions as you usually would. Important thing for these designs to work is that at the end, I randomly assign one decision that will count, so they really get the item if they have said yes or no. Okay? And so now I'm interested in the percentage of that they're saying yes or no, what I'll call the decision or stimulus value. And then I'm interested in, I also know how healthy and tasty they perceive those items to be, and how much in these different conditions they're integrating the health rating and the taste rating. So this leads me to something that looks a bit complicated, but it's actually not that complicated. This is the result from a regression analysis where I can look at the importance compared to natural, now it's the baseline, for healthiness, tastiness. You see that our participants, and this is much more pronounced in the US, so we have a mix of American and French participants, they all are making those choices based on tastiness ratings, which is the highest bar here. But what I'm interested in now is actually the way they integrate taste and health information in the different focus conditions. So these two things here, and I take this together, compute this as a factor of regulatory success. Because here what happens is that they're either integrating more the health ratings during the health condition, or you can also make a good choice, a healthy choice, if you disregard, so this is why this is negative, the tastiness of the item in the taste focus condition. These are the two paths to making a healthy choice. Right? And now what I'm doing, so the higher the score, the better they are at making healthy choices, or what I call regulatory success. And now, so I take this behavior and I ask the question, is there anything in the brain structure of these participants using a technique called Voxel-Based Morphometry that correlates with this behavior? Right? And so this gets at the idea of the density of the gray cells in those participants. And I'm specifically interested in two regions that I've written a lot about in functional studies, which is the medial prefrontal cortex and the dorsolateral prefrontal cortex. This region we know is involved in decision making, so we call it today the brain valuation system, and this is a system that is very much involved in cognitive control. And what we see is that I can take now, from my first data set, so this is a prediction exercise, I'm not going to go too much into those details, I can predict regulatory success in using the density of the gray cells, so the gray matter density, in these two regions, right? And what you see here is that my prediction is very nice, right? So it's a significant prediction that predicts very well the observed regulatory success. Then, so this is this graph here. The second graph, we have to do a bit manual animations here because the software doesn't allow for animations. So now in the second step, what we do is we ask the question, if this works in this first task, can we actually predict this also in a different sample of participants that have done a slightly different task? The task was different in a sense that we didn't tell them, oh, focus on taste or healthiness, but do anything that you want to distance yourself from the food item to make a healthier choice, right? So it's similar but not the same task. And we see indeed that we can cross-validate and cross-predict from these results to this other population, which is quite nice. So that means that there is actually something in our anatomy in the brain that can predict our ability to make healthy choices. And specifically, this is the anatomy that is involved in decision making and its control. In the next study, we have been taking this idea a bit further in a more methodological, well, no, sophisticated way is not maybe not the right term, but using a very different approach, which is a machine learning approach, where we are really trying to think about, can we develop a marker using task-based fMRI data to predict people's ability to self-control. And this here now is not in a dietary task. This is now in the monetary domain. So where I ask them to make this smaller sooner versus larger later choices. And this is something that we know actually, so this delay discounting factor that you can compute, something that also predicts, it actually correlates with obesity. It seems to be a more general transdiagnostic marker of, I would even say addictive behavior. So if you do this in addicts, you also see that this predicts whether they're addicts or not, or whether they relapse or not, if you look at these monetary decision-making tasks. And so what we know actually is that the individual differences on the structural level, like the one I showed you for the food choice task, they do exist. There are also papers that have looked at connectivity between structure and function. But what has not been done to date yet, I mean, our paper is now under review, so some ways it has been done, is the idea whether we can develop on task-based data, so while they're making those choices, a marker of delay discounting. I already told you a little bit how this would look like. So the task, I would ask Philippe, do you want 10 euros today, or do you prefer 22 euros in 14 days? Right? And what we did is, again, we are doing these cross-validation studies where we have one study with 112 participants. They're doing this task, and this is on the data set on which we train the data, and then we predict this to completely different data sets. These are German male participants. The second data set are American males and females. These are more obese, as you can imagine, right? So, and they are younger than our participants. And they do also slightly different tasks. But it's, for both of these tasks, I can measure this parameter of a delay discounting. And so what we're doing here, I'm not going too much into the details, so we can ask to the fMRI data the question, which brain regions are encoding just the decision, so the onset of the choice, which regions are decoding the amount, so the larger later amount, and which are encoding or decoding the delay. Then I throw all of this together, put this into my machine learning algorithm, and what I get is I do get actually predictive maps, multivariate predictive maps that are predicting the discounting behavior from the participants. And in some ways, these are similar regions. Well, some of them have an overlap with what I showed you earlier, to give you a bit of an idea of what they're doing. So what we see, if I would now use a meta-analysis, would look at, which regions and which of these maps are corresponding to value, reward, and effect. You see that the choice answered is related to that, and the larger later amount of delay, as the theory would suggest, are negatively linked to that. And the same is true for conflict and control, a bit less so memory and planning, because this is something that people in psychology would think that this is also linked to my memory abilities, my ability to predict myself in the future, so planning and imagery. But here we see that there's a less of an overlap with these regions in our brain marker. Now what we see though is what is interesting is that now this is the blue is the European study, the German study, the green is the American study. We do see one thing that's actually quite interesting is now we're plotting the BMI of participants, and we do see that actually in the German study there is a prediction of a relationship between the BMI and the behavior, but in the American study this is not significant. However, if we don't look at relationship between BMI and behavioral response, but look at BMI and brain response, we do find that there in both studies it seems to be the more sensitive measure, because here there is actually a significant relationship. So in other words, it seems like this marker gets at something a bit more fundamental than we would just get by running a behavioral task, which also means that it makes sense to actually look at the brain rather than just having people do this task to think about whether they are at risk for obesity or addiction or whatnot. So what I've showed you so far is that there are individual differences in self-control that can be predicted by brain structure, specifically by gray matter density in the valuation and control system in the brain. I also showed you that I can predict individual differences in self-control in the monetary domain by a distributed pattern, also consisting of similar regions that are involved in conflict, cognitive control, and also value and reward coding. And then this is more like a comment. If you are a doctor or a researcher here in the room that is interested in these types of tasks and has already data, we are trying now to take this brain marker and generalize it to other populations. Luckily, we have one study that we actually did together with Karine Clément that is here at the Institute in participants with obesity. But if you have data available to brain imaging data and delay discounting tasks, let me know. I'm very happy to collaborate and see whether I can transfer this brain signature really also to your data. That's maybe a good point also to, if you have questions.
So ma question c'est sur gray matter volume.
Yes.
Est-ce que c'est pas une variable très globale en fait donc j'y connais rien, mais ça me paraît très très global comme donnée, non?
Oui, en fait, if you want, so what in the functional analysis that I'm showing you, we are interested in kind of what is underlying this. What we're not measuring is in some ways neuronal firing. The idea is that I will see more of neuronal firing in a region if there's more density of those neurons in that region. And this is why there are studies looking at the density of the gray matters. And I think what is the interesting part is that you can localize this in the brain. So it's not gray matter density per se, it's in specific regions that you know from functional, so task-based studies that might be involved. Because the idea would be that if you have more gray matter density, maybe you can, this will make you help to activate this region more. And there's some studies that have not been done in the area of dietary self-control, but I know that, for example, Todd is working on this in Zurich. The idea, the gray matter density, obviously the brain is plastic, so I can train you. I can train you specifically, actually give you neurofeedback on a task that would increase the density in this region. So for example, in the DLPFC, the cognitive control region. And then the idea is, would this transfer that because you now have more gray matter density, you will be better performing and be more self-controlled in these tasks, right? So that's a bit the idea of why we are interested in these structure things. Today, it's true that we prefer to look at white matter connectivity, so using diffusion tensor imaging. But yeah, that's I have to come back in a year from now, I think, to to talk about the studies in that area. But it's a very good question. Thank you so much.
Est-ce que vous ajustez sur justement le niveau socio-économique ou les études?
That's the number one question that I get when I'm in this room and presenting. It turns out that it doesn't really matter for these types of tasks, right? So because we try to have participants similar to each other, so they usually have they're similar on those levels, right? But it's so the long story is it doesn't really matter for these types of tasks, right? So, but and we keep it constant in our studies because we don't have enormous sample sizes, right? So, but we obviously know that there's something linked to intelligence that helps us to make healthier choices and so forth, right? So, voilà. Now, for the second part of this talk, I will talk about something that you in some ways know much more about than I do, so I apologize. All the difficult question will be for my co-authors, right? So, which is the idea that there's actually something happening here in the gut, in the GI tract, hormone secretion, but also the composition and diversity of the bacteria, so the gut microbiome, that might be linked. That might be linked actually to decision making, right? So on a very broad level, we know that a good microbiota diversity, whatever that means, right, is actually linked to health, right? And we also know that it is influenced by what we eat, and we also know that this communicates with the brain. More specifically, this is a more complex version of this, right? So we actually know when you think about, so I'm interested in this link here, we know that in the gut or in the body, what's happening is that there are several pathways on how the gut actually communicates with the brain. Right, so there are the metabolites, there are gut peptides, there are neurotransmitters, and the precursors. There's the immune system, and then there's obviously the vagal nerve that has neuronal or neurophysiological communication with this. And so to study the influence now of the gut microbiome, again, this is research agenda that is something that I'm currently really working on, so this is really work in progress. But so we have here Fabrizio that usually uses mice. This is obviously the much better way or more clean way to study microbiome because I can have germ-free and not germ-free mice, right? So to get at causal relationships. In humans, this is difficult, right? So that's why I'm borrowing a methodological approach from behavioral economics, which is the idea I can look at changes before and after an intervention. And I'm going to show you two ways of doing this in a bunch of different studies. The first one, this is joint work again with Karine Clément's team here and Christine Patou-Bernard on we actually can look at something that gives a shock to the microbial system, which is a gastric bypass surgery. We know that after the surgery, the gut microbiome's composition and diversity is different than before. Right, so that means in these next two studies, I'll show you what actually happens on the behavior level and on the brain level if I look at people before and after gastric bypass surgery. And then the last study, this is really where we are still much more in the ongoing phase, in the data analysis phase, where instead of changing this through such a stark intervention such as a gastric bypass surgery, I do something much more subtle. I give participants a dietary supplement that is a symbioticum, so that is supposed to actually temporarily alter the gut microbial diversity and its composition. There's learning. So the first study, again, is joint work with some people here from the building, where we look at brain activity in at rest, so not during a task, just at rest, in the systems that I keep talking to you about, in the control and decision-making system. And we look how that actually predicts the change in weight, and also look at how this correlates with a hormone that's important for this, which is leptin. So here, the idea is that We know already that resting state connectivity can be different in participants with and without obesity. We also know that this can be also on a structure level, so using white matter connectivity. But our angle here is that we really try to take the connectivity at rest in those regions and predict the change in weight. And so how we did this is that we had lean control participants and 14 all female participants that qualified for bariatric surgery. Then they, so obviously only the patients, they do get the surgery, and 8 months later, all of these people come back, right? So this is the idea of this difference in difference approach that gets me some idea of causality. Obviously it's more complex, but I at least know that this has changed in between these 2 time points. And then for the patients at the CEC here, we sampled the fasting state leptin, that we can also look at this, right? So, and then we are going into this region that I showed you earlier, so this region important for decision making, and are looking for connectivity to this region, and then are looking for what you see here, whether this region actually is affected by the group membership, so whether you're patient or lean, whether it's affected by the time point, and we're specifically actually interested in the interaction between these 2 experimental factors. So what we see, what is interesting here is now the resting state connectivity between the VMPFC and the DLPFC, so decision making and cognitive control. What at the first point seems to be unintuitive is that the obese actually have more connectivity between these 2 regions at rest, because I think the layman's intuition is that, patients with obesity, they're obese because they have a problem to control themselves, and this speaks against this idea. At least it's not driven by this connectivity between these 2 regions. Right? Now what has the interesting interaction effect is actually the connectivity between the decision making and the motivation system, so the VMPFC and the ventral striatum. Here we see that the lean participants have a more connected reward and motivation system as compared to the obese, and that after the surgery, the patients with obesity have almost a similar level of connectivity in these regions. And this is also, I mean, for the layman's perspective, a bit unintuitive, because you would think, they are much more sensitive to rewards, the patients. This is more in line with the idea of dopamine deficiency hypothesis, because these are the regions with dopaminergic neurons. We're obviously not measuring dopamine here, right? So, but these are regions that have dopaminergic neurons in a higher population than other regions, and this means that maybe so this idea that the dopamine system is numbed by the participants with obesity is maybe what could explain this. Okay? And then so what we did is, we can actually take this now and predict the change in weight, right, with this connectivity. So it's a predictor, somewhere how much weight difference all the participants are having. And what is interesting now, it also, and this is obviously super exploratory because we only have it in the patients, right, but we see that this is actually linked also to the change in serum leptin levels that they're having, because also leptin before surgery, as you know, right, they're more they're leptin resistant, and afterwards they're less. And leptin is a hormone that is linked also to dopaminergic functioning. So there seems to be an interesting link here that is to explore further. Second study, very briefly. This is now a mix of these tasks that I've been talking about, where we ask people for smaller, sooner. So this is like what I showed you with my children. There's a smaller, sooner food reward versus a larger, later food reward, right? And these are also candies, like I showed you with children. And here, this is a very similar population of patients. We have them before and after the surgery. The higher those bars, the more impatient they are for food. And what we see here across 2 studies, and the tasks are slightly different, is that they are more impatient as compared to lean before the surgery. This gets to the same level after surgery, and the same is true if we do this slightly different but similar task. Same here, the higher this is, the more impatient they are for food. Significant difference before surgery between obese and lean, and then this normalizes after the surgery. So now what I showed you here is that the surgery does something that is much more than changing the size of the stomach. And again, you know much more about this than I do, what it does to the physiology, but what is interesting here is really that it changes and it improves biases in decision-making behavior. It improves their ability to make, to be more patient in reward-based choices. The last study that I would like to share is a study that was really then a brainchild of these studies with Karine, right? So because unfortunately we don't have access to the microbiome data here, so here I'm relying on the idea that there's a shock to the system, and I'm looking before after. In the study here, we have a much more complete measurement of our participants. We are putting them on a specific diet, so they eat a dietary supplement. And we know that a diet per se already can change through kind of what it acts on. So for example, tryptophan depletion diets can change impatient behavior. So these behaviors that I'm interested in. So it increases impatience for money. Here, so we're trying to get at this too and ask the question, why is this actually happening? The specific setup that we did is that we had a mix of probiotics and prebiotics. So this is also new, I think, for your type of literature, because a lot of these studies, they have looked at the probiotics. So we gave them good bacteria, 4 different ones. I'll get to this in a second. But we also added a prebiotic, so something that helps them actually to colonize. And so we could actually, this is stuff that you could buy in a pharmacy in Germany. But the manufacturer was nice enough to deliver us or manufacture for us not only the symbioticum, but also a placebo that looks identical, right? So, which is obviously important for this to work. So what I'm showing you here from the study is the same task as earlier. People make choices between smaller sooner and larger later options. And I'm interested in how the discounting parameter, so the higher the discounting parameter is, the more impatient are they, so the less self-control is changing by this intervention. And what I showed you before is, so what is interesting is that, first of all, this is for me, I always think of this as a manipulation check. So now if I look at the microbiome composition, here I'm looking at species on the genus level. Does the intervention actually have an effect? Because at the end of the day, it's not a strong intervention. The gastric bypass intervention is much stronger. Here, we're asking them, you eat whatever you eat normally, plus you eat my little dietary supplement. Right? What we see is it's not a big effect size, but we do see that indeed, after these 8 weeks or 7 weeks, they do have more of the bacteria that we give them. What was interesting, and this is data we just have since beginning of the week actually, is that this is now looking at the effect sizes of the group time interaction, is that we see these are 2 of the 4 bacteria that really are more present. But we also see that there are a lot of others that we didn't give them that are either more or less present. And I think we're now trying to figure out what their functional link is in the way they work, right? So because obviously I think it's linked to the fact that we also gave a prebioticum that there is a change also in these other bacteria. But again, this is something you have to stay tuned. What is important though is that now if we look not only at the composition, but we also look at the diversity. Here you would expect an effect on the beta diversity. We do indeed find that there is an effect on the Jacquard distance, which is a measure for beta diversity. But what is also interesting is it's really dependent, and this is similar to work from Cotillard that I think is also here in the Institute, that this is a function of the abundance of the prebiotics they had before, right? So it's an interaction between the level of the bacteria that they had before with the intervention that is really the part that is significant. And now, so Sorry, these slides are very busy, so usually I animate them. So what we also see now is this is now my measure of impatience and self-control. So we see, and this is interesting now, we see actually, given what I showed you earlier, you would have expected that they would be better at self-control and become more patient. What we find, though, is the opposite. They are actually, there is a significant difference between the groups, but those on the symbioticum become more impatient. So it's disadvantageous behavior. And we have, we tried a lot to make sense of this. It's a big protocol, so we have a lot of other measures about them. So there's no sociodemographic differences. Here we I have this formally on the slide. Also, whether they could tell, maybe they could tell whether in the treatment or in the placebo group. Now, so the belief about the treatment also doesn't matter. Also, differences at baseline on BMI and so forth, they don't matter either. And some other behavioral measures that are linked to discounting behavior don't seem to explain this. When we now look on the brain level, so remember that we have this marker. I can look at which part of the marker is actually affected by that. And what I see is that in the placebo group, there is, so when I look at the difference in time and the change in marker over time, there's no effect. But in the intervention group, so in the treatment group, I do find that the part of the marker that is involved to decoding the delay is actually affected by the intervention in the same direction as the behavior. Now, this is something that also I just got until the beginning of this week. I figured that you might be even more able to interpret this. So when I look at, so again, we have here very complete measurement of the participants. When I look at changes in tyrosine and phenylalanine, both are precursors linked to dopaminergic functioning. We also find that this correlates with their impatience. And we do see that there's slight, but these are not significant difference actually between the groups, such that for both cases, the symbiotic group actually has more of a relationship, so it has a steeper slope. And that means now, where am I going with this? So here we have, again, we have pretty much for all of these boxes, we have data. We're exploring this at the moment, and then we're trying to put this together into the model with actually the neural data in a more formal way. If you have any other ideas, I would be very interested in hearing them. So what I showed you now in this talk is that it makes a lot of sense to look also into people's neurobiology to find individual differences that could explain differences in my ability to self-control. Right? And this is also something that makes intuitively sense, right? So all of you, I'm sure, have no shows like The Biggest Loser in the US where people are losing weight, but they can't keep it off, right? And the idea that the only thing I need to do is to exercise more and change my diet, it's not an equation that is as easy as that. It seems to be there's something in our biology that makes us more or less responsive to a change in diet or a change in physical activity that help us or hurt us in keeping the weight off. And this is why I think it's very important actually to look and try to connect the dots, to look at differences in different biological markers. So here I showed you on a neural level that the cognitive control and the decision-making system seem to be important. But also that on the gut level, so the gut microbiota composition and diversity, and maybe potentially also specific hormones such as leptin are related to my ability to control my decisions. And with that, I thank you very much for your attention, and I'm looking forward to more questions.
Psychotropes can help obese people to lose weight and so to increase the reward system.
Yeah, in some ways this is a bit the next step of my research agenda, right? So what I'm really excited about is to take this knowledge that I'm gathering on the neurobiological level and help to develop something very practical. Think of an app, right, that helps you to where you can, for example, sample some biomarkers with wearables. So heart rate variability, for example, with the Apple Watch can do this. and then have through the interface certain games that help you, for example, to reduce your stress. Further research that shows that stress is detrimental also for your self-control, right? So music intervention. So this is something that's work in progress. I do think this is kind of the promise of all of this, is to go into this area of precision health, right? So where you can think about certain games or messages that would be tailored to you, right? That would help you to, to become better at self-control, or make better choices. Yeah. But that's like, you have to invite me maybe in 30 years again until I can show you this. But that's where I want to go, if I get the money to do this type of research. Yeah.
Thanks a lot for the second part. So my question is about the temporal phenomenon. So what you showed, it's before, after. So did you have the opportunity to look at longer term adaptations? Because this is really key. I mean, if we are dealing with weight loss and weight regain, we know there are short term adaptations that will not last very long. So do you have ideas about the temporal
It's a very good question, and it's a hard one, obviously, to study, right? So the first study, so the one with that was done with Karine, that took, so I didn't have any child before I started this, so it was over 15 years ago, right? So just the data collection for this took forever, right? So it's hard then to have them come back, because also participants are, there are, I can't keep them in my sample, right? So you lose participants across these data points. We are at 12 months, which is in the literature already quite nice, right? A lot of the bypass literature, as you know, right? So they look at 3 months, 4 months, right? So we had, in the behavioral protocol, we had 3 months and then 12 months. And for us, the most prominent changes were most of the time at 12 months. But we know, obviously, that then it would be very different, and it would be great to look at 2 years later. I would be excited to do this, but here we don't actually have this data, right? So, but I agree with you that this is super important, and this is very good actually to think about if you're sitting on grant committees and so forth, right? So that mostly also have short-term goals when they fund your work. That no, what we're really interested in are these long-term outcomes, right? So that would be nice to have. I'll let you know what happens with the German sample. The Germans are a bit more sticking to the rules. If I tell them, look, you need to come back, right? So they're maybe a bit likelier to stay in my sample than the French. I'll let you know. I'm German, so I can make fun of Germans, right? So, yeah.
I have the same question as Jean-Michel. Yeah. After bariatric surgery, your results are very interesting. But how long is it after the surgery? It's a short term, because it's very...
Yeah, so some of this, so the task with the candies, right, this was 12 months later. The stuff on the neural level was 8 months later, right? So yeah, it's a relative short time, right? So we have, but what is interesting, so what I did not have a chance to talk about is we also look at a bunch of different biases, so biases that food marketing companies would actually have you trap into. And we find the same pattern, that like before surgery, there's a difference between obese and lean. After surgery, this difference diminishes at a significant level. Right? So there's something really that's interesting about the surgery that's happening that goes really beyond what you, from the medical perspective, would think it would do.
And do you have any data in children, just to know at which age is?
No, not yet. I'm trying to work on this. But
it might be an evolution in time. In childhood?
Yeah, I mean, it's pretty clear that part of these decision-making systems, those are the systems that make us different from the monkeys, right? And that also are those systems that are still growing in adolescence, right? So
because, as you say, for little children under 2 years old.
Yeah, that was anecdotal, right? These were my children. Yeah, maybe they're also like very different in the way I raise them. I don't know. I can give you an update on in 10 years. So there is a developmental aspect. That's why most of these analysis we do control for age, right? So because also
it's very important in clinic use. If there is an age before we can make something for the patients.
Yeah, no, that's true. But so, and specifically, I think studies are rare in adolescents with obesity, at least in this decision-making literature. I haven't seen this, right? So, but there is there are a couple of researchers even in Paris that are looking at the latest counting task in adolescents, and that hopefully they will take me up on my offer to try to take my brain marker and try to predict the behavior in these children. And again, if you know other people that would be interested in that, send them all to me. Right? This is a big data exercise in some ways, and I really would like to have a database and a really generalize a consortium to use this also as a transdiagnostic marker. I think it's a behavior that is linked in so many disadvantageous and unhealthy behaviors that this would be super interesting actually to do.